Let's see. No, we don't really need the light. We're good. This is going to be a little annoying, but here we go. And there we go. All right, so regarding the process of transcription that you saw yesterday, it is a related process in many ways to DNA replication, but unique from DNA replication. And what makes it unique, of course, is that it is creating a different type of nucleic acid, creating RNA versus DNA. So we're going to start by taking a look at that process. So what I've got here is a representation of DNA. It's been untwisted, but not unzipped quite yet. This is located still in the nucleus, but I want to bring your attention to a couple of things. I've chosen two different colors to represent the orange for the DNA that's coding, right? And the pink, it's still DNA, but this is the promoter region. So the promoter region is something that precedes a gene. Yesterday I mentioned to you that it's kind of like when you go into a bookstore and the things that you can expect to find on any cover of a book are the title and the author's name. It's sort of like the identifiers of the minimum information that you need about that book. So in DNA, the molecular machinery that copies DNA into RNA needs this minimum amount of information. So that promoter is where the enzyme RNA polymerase attaches. So here we go, RNA polymerase attaching to the promoter, right? Now, why does it need a promoter? Plain and simple, it's a bit about size. RNA polymerase is a huge enzyme. It's a huge protein. The beginning of the DNA molecule has a start codon that's too small. So in reality, none of this is to scale, by the way. The promoter can be like 30 base pairs long. In this case, I've only got 6 base pairs. The start codon is 3 base pairs, right? 3 letters in DNA. It's a little too precise for RNA polymerase to land on, to attach to. When RNA polymerase attaches, it stimulates the separation of these strands. So what happens is you get a separation of these strands. Now, this strand up here is the 5' to 3' strand. Right? RNA polymerase, just like we mentioned with DNA polymerase in DNA replication, attaches to the 3' strand. So this strand here becomes what we call the template or coding strand. It's the one that's got the message. So this is the gene that we want to transcribe. Right? What about the other strand? This is where we're different from DNA replication. We do nothing with this strand. It literally just hangs out. Right? So for the time being, what's going to happen is we're going to start transcribing this particular strand. So for convenience, I'm going to do that on another strip of paper. I really should have found another color to make it RNA friendly. However, I have someone in the lobby, so let me admit them. There we go. All right. In any case, remember the rules for complementary base pairing between DNA and RNA. Wherever there's an adenine in DNA, there's going to be a uracil in RNA. Everything else is the same as with DNA. So as this starts moving along, right, it's going to pair an adenine with a thymine, no problem, a uracil with an adenine. The cytosine is still going to pair with a guanine. It's going to move down. Adenine still pairs with a uracil, still with a uracil, still with a guanine. Guanines will now pair with a cytosine, thymine with an adenine, cytosine with a guanine, guanine with a cytosine, thymine with another adenine, and so on until it reaches the end of the gene. And again, this is by no means long enough to represent a human gene. This is very short. Now remember yesterday we talked about how DNA is a language written in 4 letters, but all of its vocabulary is composed of 3-letter words, right? We have to keep that in mind, right? So what has happened is we've kind of, like, because this is a gene fragment, it's not meant to represent a full gene. This is sort of trailing off. It doesn't have the correct sort of complement of words, right? So don't worry about that quite now. When it does reach the end, imagine that this has reached a stop codon. When it reaches the stop codon, RNA polymerase simply releases, right? Bye-bye RNA polymerase. The RNA releases also, come on, and the DNA reanneals, so it comes back together. Now here's, and after it comes back together, it would twist back into its helical shape as well. Remember the analogy we used in class yesterday, that this is kind of like copying out a recipe that you want to, prepare at home. So you're taking your copy to your kitchen, right? So the original cookbook, in other words, the recipe and all the other hundreds of recipes that are in that cookbook, we're done with it now. It stays in the library. So this is going to stay in the nucleus of the cell, right? And you are taking this to your kitchen, which is in your home. So in reality, this is now messenger RNA, mRNA. For convenience and for length, I didn't include any introns or exons. So imagine that this has already been edited, right? So this is all good code. In other words, all based on exons. So this is all coding DNA. Now, if you're, let's say, yesterday the analogy I used was a cake. So the cake that we're going to bake is going to need certain equipment. You're going to need a baking pan, you're going to need mixing bowls, a whisk or an electric beater or a stand beater or something, stand mixer or something like that. All of this equipment that you need to actually make the product, which is going to be a protein, right, is actually in the cell cytoplasm. So the cell cytoplasm has all of these amino acids available, and it's also got lots of these ribosomes. You might remember ribosomes from grade 9 and 10. When you're drawing a cell, you always sort of saw them as tiny little dots. When you looked under a microscope, you saw them in the cytoplasm of a cell, and they just looked like grainy little dots in the background. There's actually a little more to them. They're kind of like an upside-down snowman. They have 2 sections to them. They've got what's called the large subunit and the small subunit, and they're kind of this roughly ovoid kind of shape, and they come apart. They can be brought back together, but they do come apart. For simplicity, in reality what would happen is the 2 halves would clamp onto the mRNA. For simplicity, and to give you the idea of how this sort of works its way through the ribosome, I've created these slots. So here we go. Here's our first triplet, AUG. Now, there has to be a way of delivering these to this. Right? And that way, or that structure, is something called a transfer RNA. So I'm going to tell you a bit about transfer RNA before we dive into this. Transfer RNA is the weirdest shape of ribonucleic acid. It's shaped kind of like a clover. And if you've watched any of the videos that are provided for you on the hub, you might have seen this already in the comparison. And they've got one area which is called the amino acyl end up here. And then they've got all along it, it's RNA, so it's got a code of adenines, guanines, cytosines, and uracils all along it. But it's single-stranded. So if we follow this line that I drew, it is a single strand of RNA. Right? So I'm really cheating with this now. And I'm not going to write in the letters just in the interest of time. There's a couple of spots where the letters, I will write these in, where the letters are complementary. So for example, you might have an adenine here, and a uracil here, a cytosine here, a guanine there, a uracil here, an adenine there. So what that causes is you get this sort of bonding upon itself. It creates base pairs in these three spots, and that's how you get this single-stranded RNA to assume a clover shape. Right? So imagine taking a string and just sort of like tying it together in three spots. You would get three loops, right? And these three areas that are sort of tied together. Right? Now, in these loops, the coding continues. Right? So down here, there's going to be a special triplet, right? Or special codon. So we call, by the way, to get the terminology right, these three-letter combinations that we have in mRNA, we call these codons. So each one of these is a codon. Here we have an anticodon. And there is an anticodon for every codon in the genetic code. Right? So they are complementary as well. So if your first codon is AUG, what needs to pair with it are its natural partners. So this is going to be UAC. So UAC. And that is now a tRNA. This is a delivery system for bringing in amino acids. Now, these codes, the codons, were figured out by Crick and Gamow, Crick of Watson and Crick fame, and they figured out, and I think it was also Ochoa and Nierberg, that figured that out as well. They figured out the genetic code. Right? So this is the genetic code. You've got this on the hub. You can download it in full color like this. It's provided often in a chart like this. The genetic code shows all the different codon combinations and what amino acid they stand for. Some students prefer using this one. I personally dislike this one. I've always disliked this one ever since I was a student because I find that when I'm nervous and doing a quiz or a test or something, I mess up on this. So how do you read this one? It's all about matching up the first, second, and third letter of a code. Right? So for example, if we had a codon that was GGC, we would find first letter G, second letter G, right? And then match up the row and the column. Here they are. And then C, GGC is right there. That stands for glycine. Right? If you've read through the section on the hub, you already know this, but most of the amino acid abbreviations are the first three letters of the word, with a few exceptions. The codon format, the genetic code, format that I really like is this one, the circle code. You read it from inside out. So that same codon, GGC, if I wanted to interpret it, all I got to do is find the G quadrant, GGC, there it is, glycine. Right? So using this code, it's going to be off screen, but I have to refer to it. If I have AUG, that's going to require methionine. Right? So for the purposes of illustration, I'm going to show an a tRNA bringing in the methionine. So the amino acids attached to this end of the tRNA, the amino acyl end, right? And they bring this together, right? And then the methionine is sort of in place. Now, my ribosome is a little small. In reality, 2 codons fit at any given time, right? So 2 codons at a time are in there. That's important because with one amino acid, I can't create a peptide bond. I'm trying to build a protein. I need at least 2 amino acids, right? So UUG, if I read that off of my genetic code, right, that is, and I'll show you that since we're getting used to this, UUG, it's leucine, right? So leucine, where are you? Here you are. Its amino acid, I know this is the wrong tRNA, its tRNA will bring it into place. But once the 2 of them are close enough together, a peptide bond forms. So we link these 2 together. And then this shifts by 1 codon. There you go. So AUG, UUG, now we've got CCA. That's going to be proline, the amino acid proline. So these will have shifted as well. So here is the amino acid that corresponds to proline. And we link these. And then this will shift over a bit, and the next codon, GCA, that's alanine. So, well, oh, there's one closer by. Let's shift this one into place. Alanine. And I'm just using random squares because honestly, like, I can't cut out all the amino acid shapes. So there we go. And then that shifts over. So that's GCA. Now it's going to be UCC, right, which is going to be serine. There we go, serine. And it shifts. And then it's going to be AUA, which is isoleucine. This is one of the amino acids that has an exception to the first 3 letters rule. And the reason for that is sort of twofold. I think when they were coming up with the rules for the abbreviations, they didn't want to use iso because it has meaning upon itself in terms of biochemistry. It's usually the prefix involved in some sort of isomer. But at the same time, they wanted to sort of show that it's connected to the amino acid leucine, right? So isoleucine, it's an isomer of leucine. That's all it is. AUA, GCU is our final codon here. That's going to be another alanine, right? So the code is repetitive, or as we say in biology, it's redundant. In other words, there are 20 amino acids, right? But there are more than one way of coding it. So for example, alanine, it can be GCG, GCA, GCC, GCU, right? At the same time, we can also find some that have very different sort of codes. So for example, leucine, CUU, CUC, CUA, CUG, and then it's up here as well, UUG, UUA, right? So there are 6 different codons for leucine, right? And then there are the start and stop codons. So because I created a sort of random DNA fragment, it doesn't end, right? It would keep going, and eventually we would reach a stop codon. At the stop codon, the mRNA literally is released from the ribosome. The 2 units of the ribosome come apart, and then we would have a finished protein. Now, this is not a finished protein. This is a polypeptide. Why? Because it's only 7 amino acids long. That's tiny. And the tape that's holding it together, those are the peptide bonds. So remember, 7 amino acids are held together by peptide bonds. This is a polypeptide. But it would be on its way to becoming a whole protein. Right? So the stop codons that would be at the end of a gene, right? So you have the promoter at the beginning of a gene, just refresh our memory, right? Its main role is to signal to RNA polymerase where, and to the transcription factors, where to land. Start here, that's their role. The start codon is there, and because this is only a fragment of a gene, it would keep going. I don't have a stop codon here. There would be one at the end of the gene for sure, right? What's interesting is that the start codon, unlike the stop codons don't code for anything. The stop codons are UGA, UAG, and then UAA. That's what they are. The stop codons literally don't have an amino acid equivalent. That's it. They're just stops. And that's, scientists feel that this is how they evolved, basically because you kind of didn't have a tRNA that corresponded to these. Everything else has a tRNA. So for example, phenylalanine, it's coded with UUU, right? Its tRNA anticodon would be AAA, right? This is something that is sort of automatic in terms of the binding, the complementary bonding, but you don't have that, right? You don't have it for these. The start codon, though, does double duty. It's always methionine or methionine, both pronunciations happen. So methionine, this is the beginning amino acid of every protein, so AUG. Now, whether it stays or not, proteins sometimes get modified after they are made, after they're synthesized in the cytoplasm. Proteins need further modification, because if you remember protein structure, we've now created the primary structure of the protein. That's it. So a chain of amino acids. This is the primary structure. We now have to twist it or bend it into the secondary structure, alpha helix, beta pleat. We may bend and twist it further based on polar interactions or hydrogen bonding. This is going to be the tertiary structure. And then we may actually link together different chains, like several proteins together, to make a quaternary structure. In order to do all of that, you get some of the cell's organelles involved, the Golgi body, for example, the endoplasmic reticulum, which is where a lot of this happens. Some of the ribosomes that will transcribe mRNA, messenger RNA, right? Some of the ribosomes that transcribe mRNA are attached to the surface of an endoplasmic reticulum or attached to the surface of the Golgi body, the Golgi apparatus, and transcription happens directly on the organelle, and the mRNA enters the organelle right away. It starts getting processed right away. To refresh your sort of memory, if you think back a couple of lessons, we mentioned something called the Central Dogma. And the Central Dogma states that DNA, so let's say information encoded in DNA as genes gets transcribed as RNA, in this case specifically messenger RNA, that's its job. And then this gets translated into protein. Because this represents the beginning of a protein. So when we're going from DNA to RNA, that's transcription, right? Remember the analogy I used was copying it out. So if you're copying out a recipe from a cookbook, the cookbook has been professionally printed. That's, professional ink on professional paper, and it's been bound in a book. You're copying it out with pencil and paper, right? but the message is accurate, or at least I hope it's accurate when you're copying it out. So you're copying out the message word for word, letter for letter, but in a slightly different format. That's what we're getting in transcription. So when we go from DNA to RNA, we're still staying within our idiom, right? We're still staying within nucleic acids, right? Yes, this one has slightly different chemicals, deoxyribose versus ribose, thymine versus uracil, you name it, but it's still words on paper, right? If you're thinking of the recipe analogy, it's still a way of coding information, right? When you take that recipe home, what you want to make is a cake, right? And a cake is most definitely not words on paper. A cake is yummy deliciousness of flours, eggs, butter, sugar, you name it, right? So the protein, when we get to this, we're sort of changing biochemical languages. We're going from nucleic acid to protein, right? So this is a total shift. It's something that's chemically totally different from the other two things, right? Protein is very different from that. It's amino acids, it's polypeptide. This is now a shift. So this is like translating a document from one human language to another language. Right? So we're going from one biochemical language to another. Right? Again, this is, I know it's a lot to absorb, and it's not ideal to be doing it this way. This is our reality. I'm hoping that I can post this video later this evening on the Hub. There are videos available for you to see. This document, this PDF version of this, you can download on the Hub as well. The translation section that I opened does have an assignment for this evening. I mean, for the brave, you're welcome to start looking at it and trying the assignment, but I'm going to hopefully do a bit more practice with you guys tomorrow in class and then sort of turn you loose on the assignment. For tonight, what I think you should be doing is going through the video links and looking through the resources that are there. If you want a printed hard copy of this, it's available in class and you can pick it up at your leisure. All right.